This video demonstrates the procedure for photoconversion of the spiral dye FM143 to study synaptic vesicle pools in Drosophila melanogaster. First, Drosophila larvae are dissected to expose the ventral muscles, which constitute the neuromuscular junction, or NMJ, preparation. The preparations are incubated in buffer containing FM dye, shown here in gray, labeling cellular membrane. They are then electrically or chemically stimulated to induce vesicle fusion. Vesicle membrane internalized following stimulated exocytosis will be stained with FM dye. The extracellular FM dye is then washed, removing it from the plasma membrane, and the tissue is fixed. Under continuous high-intensity illumination, the dye is completely bleached, and a black color appears due to diaminobenzidine precipitation. This precipitate can then be visualized by transmission EM.
Hi, I'm Filippo Passo from the laboratory of Silvio Rizzoli at the European Neuroscience Institute in Göttingen, Germany. Today, we will show you a procedure for FM photoconversion in Drosophila neuromuscular junctions. We use this procedure in our laboratory to study synaptic vesicle recycling. Okay, so let's get started.
Working under a dissecting microscope, begin by pinning a Drosophila larva, dorsal side up, in a Silgard-coated dish. Cover it in Drosophila saline. Using a syringe needle and fine dissection scissors, section the dorsal side longitudinally. Then remove the internal organs. Next, stretch the preparation and again pin it to the Silgard dish.